Shri Gurubhyo Namaha. I am happy to welcome you all for our online teaching academy Ekalaiva. Class 10, subject biology, unit 16, plant and animal hormones. In the previous class, we discussed about the types of plant hormones. There are 5 different types of plant hormones. Number 1, auxins, cytokinins, gibberellins, abscisic acid and ethylene. Among the 5 plant hormones, the first 3 promote the plant growth. That means auxins, cytokinins and gibberellin promotes the plant growth, whereas abscisic acid and ethylene inhibit the plant growth. Now we are going to discuss about auxins in detail. Auxins were the first plant hormone discovered. Auxins are produced at the tip of the stem. Auxins always produced at the tip of the stem and roots, from where they migrate to the zone of elongation. Once the auxins are produced at the tip of the stem as well as the root, they migrate to all parts of the plant body, especially the zone of elongation. Charles Darwin in the year 1880 observed unilateral growth and curvature of canary grass coleoptiles. Coleoptiles is nothing but a sheath of plumule. Plumule is nothing but the initial stage of shoot system, from where the stem is developed. Later on, Charles Darwin came to the conclusion that some influence was transmitted from the tip of the coleoptile to the basal part. This influence was later identified as auxins by scientist Wendt. Okay, the major hormone auxin, it is produced at the tip, it slowly transmitted to the basal part, and this influence was later identified as auxins by Wendt. Here, Wendt experiment. Fritz Wormold Wendt in the year 1903 to 1990, a Dutch biologist, demonstrated the existence and effect of auxins in plants. He did a series of experiments in Aviana coleoptiles. Aviana is nothing but a plant. In his first experiment, he removed the tip of the Aviana coleoptile. Initially, he cut the tip of the Aviana coleoptile, and then if we cut the tip, the plant will not grow. Once the tip is removed, the tip did not grow. The cut tip did not grow, indicating that the tips produced something essential for the growth. See the picture here. This is a plant. In the first experiment, Wendt cut the tip of the coleoptile. This is a coleoptile. Coleoptile is nothing but plumule, the sheath of the plumule. Initially, in the first experiment, the scientist cut the tip of the coleoptile. Once he removed the tip of the coleoptile, the plant, the plumule will not develop, the shoot will not develop. In his second experiment, he placed the agar blocks on the decapitated. Decapitated means removal of the tip. Tip is nothing but head portion of the small plant. Okay, if we remove the tip, it is said to be decapitated. So removal of tip. Once in his second experiment, he placed the agar block on the decapitated coleoptile tips. The coleoptile tips did not show any response. See here. You just keep the agar block on the tip of the coleoptile. The tips did not show any response. In his next experiment, in his third experiment, he placed the detached coleoptile tips on agar blocks. Think that it is agar block. He placed the tip here. He placed the detached coleoptile tip. This is detached coleoptile tip on the agar block. After an hour, the chemical which is present in the tip of the coleoptile, it slowly comes down and it is deposited on the agar block. After a period of time, something which is present in the tip of the coleoptile comes down and it is placed immersed in the agar block. See here, after an hour he discarded the tip. After an hour he just discarded the tip, removed the tip and placed this agar block on the decapitated coleoptile. After an hour, you just remove the coleoptile tips and now the agar block have some substance, some influence which is present. Okay. After a period of time, he removed the agar block. Now the agar block have some influence and he placed this agar block on the decapitated coleoptile. See the picture here. This is a normal germinating seed and this is called the coleoptile. Once he cut the coleoptile, the cut tip of coleoptile is removed and he placed this coleoptile tip of coleoptile on the agar block. After an hour, he just removed the tip of the coleoptile and he placed the agar block on the germinating plant. Now the leaf that has been bent, it shows some response. It grew. It means that coleoptile plant shoot system, plumule will grow. It grew straight up, indicating that some chemicals had diffused from the cut coleoptile tips into the agar block. Very, very important. Some chemicals which is present in the tip of the coleoptiles diffuses into the agar block. Once we place the agar block on the plant shows response. From his experiment, Wendt concluded that a chemical diffusing from the tip of the coleoptile was responsible for the growth of the plant and he named it as Auxins. He named the chemical as Auxin. That means Auxin means to grow. So finally he concluded that some chemical which is diffused from the tip of the coleoptile to the agar block. This agar block on placed over the plant shows response. Finally he concluded that some chemical was responsible for the growth of the plant and he finally named it as auxin. The word auxin means to grow. Clear, children? Then types of auxins. Auxins are classified into two types, and namely natural auxins and another one is synthetic auxins. The name itself you can easily understand. Natural auxins. These auxins are produced by the plants. So auxins which are produced by the plants, then it is said to be natural auxins. Example, IAA, indole 3 acetic acid. So IAA is a good example for natural auxins. This auxin are produced by the plants itself. The second one is synthetic auxins. Artificially synthesized auxins that have the properties like auxin, then it is said to be synthetic. Synthetic means artificially prepared auxins. So man-made auxins, it is prepared in the laboratory. Artificially synthesized auxin have the property of natural auxins, then they are said to be synthetic auxin. Example, 2,4-D, 2,4 dichlorophenoxyacetic acid. So it is a good example for synthetic auxin. These auxins are artificially prepared in the laboratory and it has the property of natural auxin. Clear, children? The next one, physiological effects of auxin. Auxins brings about variety of physiological effects in different parts of the plant body. You know very well, auxin is a plant hormone. It is mainly synthesized at the tip of the shoot as well as at the tip of the root. It plays a vital role in the physiological effects and it plays an important role in the plant body in various parts of the body. So first, physiological effects. Auxins promotes the elongation. We already discussed about elongation. It promotes the plant growth. So auxin plays a very important role in the elongation of stem. It promotes the elongation of stem and coleoptiles, which makes them to grow. So the main physiological effect, the first one is auxins promote the elongation of the stems. Second one, auxins induce root formation. It induces the root formation. It is usually present at the tip of the roots and it induces the root formation at low concentration and it inhibit it at higher concentration. If the auxin level is higher means it inhibit the root formation. When it is low concentration means it induce means stimulate the. Stimulate or promote. So when the level of the auxin is in low concentration, it promotes the root formation. If the level is in increased high concentration, it inhibits the root formation. Clear? A third physiological effect, auxins produced by the apical buds. Apical buds, apex means at the top. Auxin produced by apical buds suppress the growth of the lateral buds. This is called apical dominance. It is very important, 2 mark question. What is apical dominance? Apex, the auxins which is produced at the tip dominate the entire plant. Okay, so the apex, the auxins produced by the apical bud suppress the growth of the lateral buds. The apical bud suppress the growth of the lateral bud. That is called apical dominance. Fourth one, seedless fruits. There is no seed at all. Seedless fruits without fertilization. So if there is no fertilization means that process is called parthenocarpy. Parthenocarpy means without fertilization. So seedless fruits without fertilization are induced by the external applications of auxins. Example, watermelon, grapes, lime, etc. The fifth physiological effect, auxins prevent the formation of abscission layer. What is the meaning of abscission? Abscission means shedding of leaves. removal of plant parts from the plant. What are the parts? Leaves, flower, fruits, etc. So it prevents the formation of abscission layer. Clear? Thank you children. We will see in the next class.